Okay guys, so in this video we're going to talk about healthcare biotechnology. um, Biotechnology is helping us to heal the world, healing by harnessing the nature as toolbox and using our genetic makeup to heal and guidelines research by reducing the rates of infectious disease, changing the odds of serious and life-threatening conditions affecting millions around the world. Maksudnya we have better treatments, and then we can also tailoring treatments to individuals to minimize health risks and side effects. So customize treatment, but because not everyone has the same, though we have the same organs and the same structure biological system, but the way our biological system from one another can be different. So our reaction to drugs or treatment can also be different. So healthcare biotechnology help us to customize, to tailor the treatment based on that individual's genetic makeup, and then create more precise tool for disease protection and combating serious illnesses. um, Healthcare biotechnology is just an application of biotechnology to diagnose health and illness and treatment of human disease. Okay, so if this is the definition, so there are many, it's easy to see how biotechnology can be used for medicinal purposes, based on the knowledge of our species and the genetic basis of heritable diseases, and the invention of technology to manipulate and fix mutant genes provide the gateway, provide the methods to treat diseases. These include making designer drugs. We can use computer modeling to design drugs without the lab protein structure. Maksudnya kita just model dulu guna komputer. We don't really have to structure the drugs inside the lab to manufacture the drugs first. So kita just tengok dulu melalui computer modeling, apply dia dekat animal model system, for example, and tengok whether it's working or not. If it's not working, then it's pointless to manufacture the drugs in the lab. But if there is a possibility to for the drugs to treat potential untuk di-develop dalam lab dan di-commercializekan for clinical trial pula. Okay, to see whether it's working or not dekat human sample. And then we have replacement therapies. This is to treat the lack of production of normal substances. For example, insulin. Diabetic people, they can't produce insulin, so we replace by replacement therapies. Kita replace balik dia punya hormon insulin tu melalui injection lah, right? Sama juga dengan orang yang who cannot ada masalah blood clot, tak boleh nak clot darah bila ada luka, and then kita akan bagi dia clot factor bila dia bleeding. Then we also have gene therapy, and then the replacement therapies and the gene therapy, they are basically to replace something, but the difference between these two is that the replacement therapies kita actually replace the protein. Insulin, clot factor ni semua adalah protein. In gene therapy, however, we actually replace the genes. Maksudnya kita pergi terus dekat nucleus of the cell, and then kita repair DNA tu terus. Okay, contohnya dekat bahagian ni adalah and then kita remove the defective genes, non-functional one, dan kita bagi dia normal insulin gene sequence, right? And then if berjaya, then bila this gene is being transcribed first into mRNA, mRNA, and then translated into proteins, So that patient can already produce dia punya insulin balik. Okay, so kita dah fix it. No need to do replacement therapy. Contoh ni secara theoretically, and untuk bezakan apa maksud replacement therapy dengan gene therapy. And then we also have immunosuppressive therapy. This is to inhibit rejection. This is especially bila kita buat organ transplant. Contoh, when two people are doing liver transplant, seorang donor, seorang adalah recipient of the liver cells. Tak semestinya walaupun ada compatibility, kan kita usually doctors will check whether sesuai atau tak untuk the recipient to receive liver organ from the donor. Okay, they will check. But then again, when you do the surgery, when you do the transplant, tak semestinya the recipient, the badan, the immune system in the of the recipient can accept that outsider punya cells. Okay, so sometimes badan kita akan ada autoimmune response to benda yang masuk dalam badan. Okay, so how do they actually want to solve this problem atau reduce this immune response is by doing this immunosuppressive therapy. So maksudnya dia actually lowerkan you punya immune response. It's quite dangerous kalau berlaku infection lah. But then again, they know how to handle this condition, right? And then we also have cancer therapies using antisense technology. If you remember our flavor saver tomato, which actually use the antisense technology to inhibit the translation of the PG enzyme. So konsep yang sama dekat sini, what they do is that they actually identify dekat mana cancer mutation tu. Cancer tu disebabkan oleh mutation of genes dekat bahagian mana dekat kita punya genome. Right? And then we create a complementary mRNA. Right? Complementary mRNA to that defective gene, to that mutated gene yang give rise to the cancer, so that bila berlakunya transcription and then translation of that part of that mutated gene, our complementary mRNA can bind to the mRNA of that mutated gene. Right? And then bila dia combine, you know what happen lah. Translation tak boleh berlaku. Okay, so this is the mutated gene punya mRNA, for example. And then this is our mRNA yang complementary to that mutated gene. So bila dia complementary, dia akan bind. They form a hydrogen bond and they bind. Dia jadi double stranded DNA balik. So when they are in this form, ribosome dekat cytoplasm, they can't read this thing, so they can't translate this thing. So bila dia tak boleh translate, dia tak akan menghasilkan the cancer protein will not be translated, will not be produced. And then this actually, bila the formation of double stranded DNA in the cytoplasm will actually trigger enzyme, I think a dicer enzyme will come and actually degrade this thing. Dia akan gunting dia into pieces. Habis kat situ.